Welcome to the last video on the unit for bacterial growth and nutrition. In this video, we're going to focus on looking at binary fission, how bacteria actually grow, and studying the exponential growth rates of bacteria. This is going to involve doing a little bit of math and calculations to be able to determine how many bacteria might be in a sample after a certain amount of exponential growth has occurred. Let's go ahead and get started. All bacteria replicate through a process called binary fission. This is an asexual reproduction mechanism where one parent splits into two daughter cells. Conceptually, it's similar to mitosis in eukaryotic cells, but it's a simpler process. This is because bacteria don't have a nucleus, so they don't have to worry about removing the nuclear membrane before dividing. Binary fission consists of two main processes. The first is DNA replication, where both the chromosome and any plasmid DNA that are in the cell are going to replicate, so there's two copies. The second stage is going to be cell elongation, where the cell itself is going to start to get bigger. In order to grow in size, the cell must be synthesizing all of the components of the new cell, including additional peptidoglycan as the cell wall is stretching, proteins for metabolism, and ribosomes and other internal structures such as inclusion bodies. As the cell elongates, it actually drags one copy of the DNA towards one side of the newly replicated cell, and the other copy towards the other, thus separating the copied genomes from each other. The elongated cell is then cleaved by the formation of a division septum. This consists of specific bacterial proteins that invaginate the cell membrane in the cell wall and eventually pull together until they fully separate the two cells. For species that tend to form specific arrangements, such as streptococci that form chains, the two separate cells do not necessarily release from each other and instead remain attached, forming the chain arrangement that's characteristic of that species. Because binary fission is asexual, there's no combining of different genetics from two different parents. Instead, a lot of the bacterial genetic diversity in a population comes from mutations that happen in the DNA during the replication process. When bacteria are grown in a laboratory under optimum conditions, they undergo what is called exponential growth. This means that their numbers double with each round of binary fission. In other words, one cell will become two cells. Those two cells then become four cells, which all replicate and become 8 cells, and so on and so forth. The time it takes for this doubling is called the generation time. And you can calculate it with the equation that generation time equals time, or t, divided by the number of generations, represented by n. Keep in mind that the number of generations just means how many times has the cell divided or undergone binary fission. For some bacteria, such as E. coli, the generation time is as fast as 20 minutes. This means every 20 minutes the cells have divided. This is the reason that some species grow so quickly, and you can inoculate a broth culture with a few E. coli cells, and by the next day you will have billions of cells in your tube. However, not all organisms grow quite that quickly. The generation time for Mycobacterium tuberculosis is about 16 hours, and it would take a full week to grow a liquid culture in the lab. Because a specific species of bacteria will grow at a set rate, you can actually calculate how many cells will be in your culture after a given period of time. This is done by using the exponential growth equation, which is seen here, where little b equals the number of cells after growth, big B is your number of cells before growth, and then again, n is the number of generations. Let's take a look at how we use this equation. Here is an example problem that tells us that we have 50 bacterial cells in our sample, and that this particular species has a generation time of 45 minutes. It then asks, how many cells are we going to have if we allow those 50 cells to grow for 4 hours? So remember, the equation we're using is little b equals big B times 2 to the n. with little b being the amount of cells after growth, which is what we want to know. How many are we going to have after 4 hours? So right now we're trying to solve for little b. Big B was the number of cells before growth, which in this case was 50. But then when we go to fill in N, notice the problem doesn't tell us the number of generations. Instead, it tells us the generation time. So besides just this equation, we also have to use that the generation time equals the time of growth divided by the number of generations. Now here we have 45 minutes for the generation time equals they've been growing for 4 hours, and we're trying to solve for N. So we rearrange this equation to get N equals 4 hours over 45 minutes, but then we have to make sure our units match. And so we can't have hours divided by minutes. There are 60 minutes in an hour, so 4 times 60 is 240 minutes, divided by 45 minutes gives us an N of 5.3 generations. So during this 4 hours of growth, the cells have divided 5.3 times. Now we do use the 0.3 in this number because although one cell couldn't go through 0.3 of a replication, the entire population is replicating. And 0.3, or 30% of the population as a whole, could have finished their replication during that 4 hours of time. So now we know our N. We can take N and plug it back into our original equation, so 2 to 5 to the third. And when we solve for that equation, it's going to give us little b equals 1,969.8 cells. So after 4 hours of growth, we would have 1,969 cells that are in our culture now. It's important to note that when growing bacteria in the lab, they do not go through this exponential type growth for the entire time. There are distinct phases of bacterial growth when they are grown in a closed culture, also called a batch culture. A closed culture is the main way we grow bacteria in a diagnostic or research lab. Closed culture just means that we don't continually feed our microbes and we're not continually removing their waste products. So eventually, food is going to decrease and become limiting, and the waste products are going to increase. This flask here is a standard example of a closed culture in a bacterial lab. When bacteria are inoculated into a closed culture, meaning the original cells are first added to the media, they have to adjust to their new environment. This adjustment typically involves things like transcribing and translating new proteins, performing different metabolic reactions based on the food sources in the media, and beginning the process of binary fission. This first stage is called the lag phase, and during it, there's no actual binary fission that is happening, so the number of cells does not increase. The next stage is the log phase. This is when bacteria begin going through binary fission, and they start their exponential growth of population numbers. It is only during log phase that we can use the exponential growth equation that we just learned on the last slide. The next stage of growth is the stationary phase. During this stage, the bacteria have started to run out of their nutrients and waste products have started increasing, so cells are starting to die. However, new cells are also still forming through binary fission. This means that the overall number of cells becomes static. The number of cells isn't increasing or decreasing because for every cell that dies, a new cell divides and a cell takes that dead cell's place. Those dying cells are actually going to become the nutrients for those that are still living and dividing. However, eventually the number of cells that die starts to outweigh the number of cells that are dividing, and the overall population numbers will decrease during the death or decline phase. If this line were to continue out over time, you'll find that the line will eventually level off without actually dropping down to zero. Even in a closed system, some bacteria will continue replicating and supporting the population, with the dying cells providing the additional nutrients. If you don't want the bacteria to go through all stages of bacterial growth in the lab, you can grow them using a continuous culture instead. During a continuous culture, there are ways to add nutrients to the cells and remove waste products. By controlling the amount of nutrients and waste products that accumulate, you can keep the cells in either continuous, exponential, or stationary phase growth. This is typically what's done in industrial microbiology, where the microbes are being used as little biological factories to produce molecules for us. For example, insulin-producing E. coli are kept in exponential phase so that they're continuously transcribing and translating the insulin protein, which is then purified from the culture. Other products, such as antibiotics, are produced by cells during stationary phase, and so the cells would be grown under conditions that keep them growing in a stationary population instead of an exponential one. The equipment to maintain a continuous culture is called a chemostat. Both closed cultures and continuous cultures represent ways that bacteria grow in the laboratory. However, neither is representative of the way bacteria grow in the environment in nature. Most environments have limited resources such as nutrients, and they have changing physical environments, which prevents the populations from following standard bacterial growth curves that we see in the lab. Interestingly, many bacteria actually grow as biofilms in nature instead of as planktonic or free-floating cells. Biofilms are communities of bacteria, typically many different species, that actually grow as a community or ecosystem. These bacteria form solid masses on surfaces. The cells in biofilms actually secrete something called extracellular polymetric substances, which form a sticky layer that is difficult to penetrate. This means that biofilms are difficult to destroy with antibiotics or chemicals because the molecules just don't get to the cells that are buried down under the biofilm itself in that EPS. One biofilm you're probably pretty familiar with is the plaque that's found on your teeth. This is why dental hygienists actually need to go in and scrape when they're cleaning plaque. They can't just use a chemical or something like Listerine to remove it because that biofilm is stuck onto the enamel of the teeth and those cells are protected from chemicals like those that are found in Listerine. Biofilms will also grow on rocks, in streams, on soil particles, or on various tissue surfaces. In the medical field, biofilms can be problematic because they form on solid surfaces that have been inserted into the body, such as catheters or joint replacements. And when this happens, standard antibiotic therapy can't destroy the biofilm, and so typically you have to go in surgically to clean out those joints. Biofilms form in a stepwise manner. First, the planktonic cells start to lightly attach to the surface, but this is transient, and the cells will attach and come off and attach and come off. But eventually, a few become more permanently stuck to the surface, and those will start to undergo binary fission, leading to growth of the population. Once the population gets to be a certain size, the cells will start producing and secreting the EPS, and the biofilm structure will start to develop. The cells are able to know how big the population is by using something called quorum sensing. Quorum sensing is a way that bacteria can communicate with each other to count their numbers. The cells produce a chemical signaling molecule that doesn't do anything at low concentrations, but as the number of cells increases, the amount of chemical being produced increases. Once it gets to a high enough concentration, it binds to receptors on the surface of the bacterial cells, leading to a change in gene expression within the cell itself. In this example, the chemical signal causes the cells to start producing the EPS molecule and secreting it out to form the biofilm. The biofilm is a complex structure. It consists of embedded cells as well as channels for water and nutrients to be shared among the cells themselves. Once the biofilm gets to be a certain size, some microbes are going to break off the surface of it, becoming planktonic cells again, which will then disperse to other locations in the environment where they can start the biofilm formation process all over again.